Outcome 1, Gregor Mendel. Who's he? Father of what? Genetics. Discover the traits are inherited through discrete units called alleles. His principle of segregation, dominance, and independent assortment, which are his 3 principles that you must know for the exam. Explain how traits separate and combine in offspring. For example, pea plants with tall, capital T, and short alleles demonstrate dominance, where tall T mass short t. Can we just be very clear on the difference between gene and allele? Okay, what is the difference? Okay, so a gene codes for a trait, while an allele is a different form of that trait. So if we look at the plant example given, the gene is for height of the pea plant, right? And the allele is either tall allele or short allele. Okay. So dominant masks the effect. How many dominants do you need to be expressed? 1 or 2. If it's recessive, you need 2 little t's, for example. Okay, independent assortment is genes for different traits separate independently. And segregation is alleles separate during gamete formation. Okay, so what's the difference between those 2 things? So dominance was that one would be dominant over the other. That's his first law of dominance. The second law is the law of segregation, that alleles separate during gamete formation. So that means what? Like if we're talking about sperm, for example, right? One sperm. So if a person is heterozygous, we know that one sperm would get the big T and another sperm would get the little t. So they alleles separate. Independent assortment is that genes of different traits separate. So the chromosomes will randomly go to whichever side of the cell. Does that make sense? It doesn't matter if you're blue-eyed and red hair, that both of those traits would go that way all the time, right? So your hair color and your eye color don't necessarily go unless they're attached to the same chromosome. That's different. Okay, but Mendel didn't know that. Okay, number 1, what does Mendel's principle of segregation state? A, B, C, or D? A. Okay. In pea plants, tall alleles dominant over short with phenotype will a plant with genotype big T, little t show? Tall height. Remember, it's not a blending of traits anymore. Okay, so any questions about that terminology? Okay, so please know Mendel's 3 laws. It'll probably state Mendel's 3 laws, so put a star there for 3 laws. Number 2, I feel like most of you, almost all of you know this one already. Heterozygous, homozygous. There are 2 types of which one? Homozygous or heterozygous? Homo. So what are they? Homo dominant and homo recessive. We're using the T's. What would homo dominant look like? Big T, and homo recessive would be little t, and heterozygous will be a blend of big T, little t. It's terrible to write up. Okay, a carrier. Do you remember a carrier when we did pedigrees? Okay, so they carry the trait, but it's not being expressed in their phenotype. Okay, so which term describes an organism with 2 identical alleles for a trait? Hetero, homo, hybrid, or carrier? B. If an organism is hetero, which statement is true? It has 2 identical alleles, it carries 2 dominant alleles, has 2 different alleles. It is always affected by a recessive trait. Which one? C is correct. All right, get some good stuff. No. Genotype, phenotype, where are they found? Where's genotype found? DNA. Phenotype, what's another way to remember it? I always think physical, because I think pH, and genes, not the kind you wear, right, are expressed on your DNA. Okay, so if I give you blue eyes, what is that? Genotype or a phenotype? Pardon? Not good. Okay, and AA? Phenotype. What? Phenotype? Little a? Genotype. Okay, the physical appearance of an organism is known as its phenotype. And if two plants have genotypes, big T, and little t, big T, little t, their phenotypes will be what? Be both tall. Okay, so we remember that expression, right? You need just one big T or whatever, one big letter to express phenotype correctly. Let me see what's here, because I want to do some examples of crosses yet. Okay. Punnett squares and autosomal. Punnett squares are what? Yeah, so it's to show probability. It's a way to organize, right? If you're with me in chem, you'll know a rice table, right? It's the same concept of organizing or thinking, right? Please remember that if you are given a question with two heterozygous, you don't need to cross it. You know that they should field a 3-to-1 phenotypical ratio, right? You remember that 3-to-1? And what was the special one for dihybrid? 9, 3, 3, 1. And that's when you are what? Heterozygous for both traits in both people, right? Okay. Test crosses you need to know. Okay, so test crosses help identify unknown genotypes by crossing with a homozygous recessive individual. I'm going to highlight that. Well, why would I highlight that? I don't know. Okay. Okay, so you can put them in ratios or what other value? Percentages, right? So fractions, percentages. I don't think we said which type on the exam, so but do know what both mean. Okay, you know that 25% is a quarter, right? And 50% is a half. Okay. Purebred means what? Homozygous. So what are the two types of purebreds you could have? Is it you using T's again in this one? Yep. It could be big T, or little t, right? Both of those are purebreds. Okay. And a hybrid would be like a big T, little t, right? Okay. What is the phenotypical ratio of a monohybrid cross between two heterozygous? What's that, 3-to-1? Okay. What is the purpose of a test cross? B. Okay, let's do an example. I'll make one up. Let's pick an organism. Anybody? A chipmunk? Okay. Okay, so we have a population of chipmunks. I don't know how to draw them. They're going to be X's. Okay. We already determined, what did we determine about chipmunks? What's the trait we're going to look at? Do they have the white spot on the head? White spot on the head or not? Okay. Or not. Okay, which one's dominant? The white spot or no white spot? White spot's dominant? Okay, so what letter should we use? W would be terrible. A. A and little a, because you can definitely distinguish between big A, little a, right? Okay, so we randomly select a chipmunk, this one, by random. Okay, it has a white spot. We want to figure out its genotype. What would we do to figure out its genotype? So we know it's A something, right? What would we do? Okay, so we're going to do a test cross. Does that make sense? So we do a test cross to figure it out. So we take our chipmunk and what do we cross it with? Louder. Okay, so here's our chipmunk. We don't know. It can either be, which two scenarios can we have for our chipmunk that were randomly selected? Big A, or big A, little a. What are we crossing it with as test? Little a, and little a, little a. Okay, I don't know the gestational period on a chipmunk. So we'll pretend it's like super quick. Okay, so we do our cross, right? We get big A, 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 right? We do this one, and we get this scenario, okay? So what do we know about the first test cross that we did the probability of? 100% of the offspring will be what? Yeah, and we'll have that white spot, okay? Here we have a 50-50 chance, right? Of having spot, no spot. So we breed these 3 generations later, and what do we find? So we're going to breed 3 generations of this random chipmunk with our test cross, and we're going to find a 50-50 ratio of white spot to no white spot. So what can we conclude? Our original chipmunk is heterozygous, or big A, little a. Okay, so we understand how test cross works? Yeah? Okay. So you'd have to give some written explanation, which I'm not going to write on here because, fun times. Can't write on this. Okay. That kind of concludes what we did in grade 9, right? Except test cross. Now we go into non-Mendelian inheritance. Okay. Do you remember some of these words? Okay. Do you remember that we use some different nomenclature or ways to represent? Sometimes we use prime, sometimes we use different letters, sometimes we use like a big letter and then a little letter, right? Do you remember all of that? Okay. So we did lots of good problems, so that would be a good way to go back and look at those problems. So codominants both express equally. So what would I see in codominance if I'm looking at flower color? If I think of flowers. If we use flowers for all of these examples. Both alleles expressed. What would I see? Okay, so if they're blue and green, or blue and purple, blue and red. Ugh, really ugly. Okay. Okay, we have blue flowers and red flowers, and we breed them. If they're codominant, what would we see? We would see both blue and red. So some petals would be blue, some petals would be red. They're both expressed. If it's incomplete dominance, and we cross a blue flower with a red flower, what do we get? What does blue and red make? Blue and red make purple. We would have purple flowers as well. Okay, so a blending produces a new phenotype while codominance does not, if you remember that correctly. Okay, and multiple alleles, we always use the blood type idea, right? So there's 3, not 2 going on. Okay, so we have A, B, and O. Do you remember what they were when we wrote out A, B, and O? No? Okay. So if we had blood type, if I had A, what would my genes look like? Genotypes. Remember we used I? Okay, we used I big A, or I big A and little I. Remember that? Okay. B would then be what? IB, or IB, I. O is what? Little I, and which one am I missing? AB. And AB is IA, IB. What does AB show? Codominance or what's the other one? Incomplete dominance. Codominance is correct. Okay. So Snapdragon, super popular, so probably remember, right? What was the thing with the Snapdragons? You had a red one, a white one, and you mixed them together. You got pink flowers. That was an example of incomplete dominance, right? Where you make a new phenotype. Codominance, we talked about cows, roan cows. Remember that? You have a question? No, you're just okay. So in snapdragons, crossing two pink flowers produces what? Okay, so you have RW, RW. Okay, so you don't have a blending, so that's how you know it's incomplete, and you made pink. So probably the R was going to be red, and the W would have been from a white flower, right? So if you cross this, what would you get? So you're going to have an RW and an RW. We'll do the Punnett square. Oops. W. Okay, what did we get? All three colors? Okay, what percentages or ratios? 25 red, 50 white, 50 pink and 25 white. If I were going to do that in a ratio, I would have what? 1 to 2 to 1. Okay, so remember 1 to 2 to 1. 1. Okay. Which blood group system demonstrates multiple alleles? Yeah, I don't like that question. We talked about that. So go back and definitely look at that. Okay. Well, at the top of, can I answer that tomorrow? I'll look it up. Yeah, but no, because that's 11. That's the outcome for 11. Remember when I said the other day, I don't know if I said it to your group, I know I said it to Ted's. When we wrote the exams, we looked at the outcome and we made sure that we included questions on the exam that are about the outcome. So the knowing who donates to who is not a grade 12 thing you need, it's a grade 11. So that will not be on the exam.
Okay, so that will have a table to fill it out with which one was which.
Yeah, no, that's not on this exam. Okay. So that's why I put the outcomes here, because it shows you what should be on the exam. Okay. Sex determination in humans. Okay, what do we know? XX and XY are what? XX is female. XY is male. Okay. We know the Y chromosome is significantly smaller than the X, so therefore it has less room for less genes. And that's what gives us those Punnett squares that are different when we're looking at sex traits. Okay. Females are usually XX. You can have different variations, right? You can have XO. Which parent determines the sex of a human child? Who? The dad is correct. Either the dad is giving a Y or dad is giving the X. Okay? I'm pretty sure you need, this is an outcome. So you're going to have to answer a question about male, female, and XX and XY. Okay? I'll also look at that and get it back to you tomorrow. Most of them are multiple choice. Yeah. More than half, I would say, yes. Okay. Okay. Let's get into some things. Again. Okay. Sex-linked traits are often the X chromosome, affecting males more because they only have one X. For example, color blindness, sickle cell anemia, right? Sickle cell anemia, that's sex trait. Okay, so sex-linked are when it's on the X or Y chromosome. What pair is your sex chromosomes? Last one, or pair 23. Pair all 1 to 22 are called what? Autosomal. Okay. Carrier is usually a female with only one recessive, right? So she would have X big A, X little a. That makes her a carrier, okay? Because she's carrying that and she could give that to her offspring. Hemophilia, that's the one I wanted. Not sickle cell. Hemophilia is X-linked disorder affecting blood clotting. Okay? So why are sex-linked traits, recessive traits, more common in males? Yeah, so they only have one X. So if they get one chance out of two, okay, say that again, Amaris.
They don't have the second X that could be dominant, so there's nothing to mask the effect of the recessive.
Good. All right. A woman carrier for red-green color blindness has a son. What are the chances that the son is colorblind? Okay. Doesn't say what the dad is. Okay. She's a carrier though. So what do we know about the mom? Yeah. Okay, what do we know about the dad? Well, we know he has an XY, right? Okay, so what is the dad giving the son?
One.
The Y, right? So the dad doesn't really give the red-green, right? So she's going to give him one, big A, one little a. So what's the chance of the son? 50%. Good. Yeah. Is there pedigrees coming up? Okay. Right here. Okay. Oy. Pedigree charts track traits across generations. Do you remember how to draw a pedigree chart and what the symbols meant? Yada, yada. Unfortunately, we do. Okay. Circle. In the pedigree chart, what symbol usually represents a carrier female? Well, good. Okay, what is the purpose of a pedigree chart? Okay, why is that important? Yeah, you might have this because, okay, so a man and a woman have a union. Do you remember that line? This way is called a marriage line. You don't have to be married to have children, but whatever. The line down here is to their offspring. Maybe they have 3 children. You have 2 boys and a girl. The girl goes to marry and have offspring with this gentleman, and they have that. And then this gentleman goes here, and then he has this. Okay. How would I label this generation-wise? You remember that part? Generation 1, generation 2, generation 3. We also know that we can number the people in the generation. 2, 3, 4, 5, and 1, 2, 3, 4. Right? Okay. What do we got? Let's do this.
Marks for no marks in them?
Hmm?
Like marks for not marking them?
I think we should be keeping a tally. I wish I had a paper here. So I'm gonna figure out how many questions, and I'm gonna figure out if you have to draw. I feel like we didn't. I feel like you have to identify. I'm 100% sure you're gonna have to identify something off the pedigree. I don't think you're drawing. So I think the outcome was to read a pedigree, not draw a pedigree. Okay. I don't know if that's gonna work. We'll see. Okay. Hi, Ben.
In generation 2, you have to be a carrier for the daughter to have it.
I thought it was a carrier. Didn't it?
No, like over there.
Generation 2. Oh, okay. G2 person 1. Okay, so let's figure that out. Okay, so is it dominant or recessive just by looking at the pedigree? Do you remember that part? Is it in every generation?
Oh, yeah.
Yes. So therefore, is it dominant? Yes, because it's in every generation, right? Okay, so we can say that. I don't know if this one's gonna work, but let's pretend. Okay. Is it sex linked? Okay, how many? I don't know if this one's gonna work. How many males and female ratio are affected? Way more women are affected. 1, 2. Let's do this way then. Let's do that. Okay, so what do we have? We have 1, 2, 3 males and 2 females affected. Okay, so we can assume maybe X-linked. So we're assuming sex-linked. Okay, so we're assuming sex-linked dominance, right? Okay, so is it true? I don't know. I just made this up. So to have that, you would either have big X, what? This. Okay, so every female is this who has it, right? Does that make sense? Okay, every male who has it would have to have one big A and they'd have to have a Y. Okay, if they're half shaded, what would they have? What the heck was I drawing there? Draw my letters today. X big A, X little a. Okay, and right here, X big A, X little a if they're carriers. Person Gen 3, number 3 would be what? X little a. The dad here, since he's not shaded at all, would be X little a and Y, Gen 2, number 5. And what would Gen 1, number 1 person be? Okay. Now, I don't know if we're going to want to go through and see if this works. Okay, so that's how we would label it, right? Do we label everybody? Did I miss somebody?
Yeah.
I did. What is that person going to be? So if they're a carrier, then I labeled that wrong. Okay. So do we understand? They'd have to be recessive to be a carrier, not dominant. Good? Yeah, you remember how to do that? Okay. Oh, this is a long unit. Do we need a break? No, we're good? Okay. Non-disjunction means what? The chromosomes don't separate properly, right? Non-disjunction we see in trisomies, which are 21 or 18, which we commonly refer to as what? Down syndrome, right? There are 2 other ones that we looked at. Do you remember them? Edwards syndrome and Klinefelter's were the other 2. So know about those.
Oh, so it's gonna ask us about those?
I think there's one question. Yeah. So just remember that there are 4. Okay. But at non-disjunction can happen at other chromosomes, right? And it depends. The bigger the chromosome, the less, if it's someone's missing one of them, the less viable, right? Because that's where a lot of the information is stored. Okay. So non-disjunction is when they don't separate properly. Monosomy, mono means what? 1 and tri means 3, right? So if you have the correct amount of chromosomes, you're going to have 2 chromosomes. Correct? Or normal, not correct. So if you were normal in bio, you would have that. If you're a monosomy, you would have what? 1 mono. And if you're tri, you would have what? 3. Okay. So what is non-disjunction? Failure of chromosomes to separate. Okay, what is the condition caused by trisomy 21? Down syndrome. Okay. So That was pretty quick outcome on the exam, if I remember right. Okay, identifying chromosomes from mutations and karyotypes. Okay, I have a picture here. So where I started realizing I should be using pictures. Okay, so we look at chromosome 9 and we look at chromosome 22 because they're both circled. So is this a normal karyotype? Yes or no? No, because there seems to be some concerning chromosome areas, right? Normal, what would a normal karyotype look like? You would have how many of each chromosome? 2. Okay, and they would be roughly almost the same size or extremely close to the same size. If they're not, then something occurred to cause some sort of non-disjunction, some, do you remember insertion or deletion as well, to cause one chromosome to be shorter and one chromosome to be longer, for example. Okay? So, karyotype is a picture of your chromosomes arranged in what order? Yeah. Hi, Ava.
I was wondering, because there was a question on one of the things where it was like, it was kind of like that, but how do we know if it was like, wasn't there one where it was like transferred over or something like that?
You show me the question and we can look at it.
I don't.
Yeah. Okay. I'll think through that question. Okay, so number 9, obviously the chromosomes are of different lengths. So what can we assume here? That part of this chromosome attached to the other one, right? And 22 here, part is missing. We don't know where it went. It was deleted, right? Okay. So karyotype showing XXY indicates what type of condition? It's Klinefelter or Turner. I can't. Sure, it's Klinefelter. Turner Superwoman. Yeah. Okay. Oh, it does? Oh. I didn't think it. Okay. What does monosomy mean? One missing chromosome. Yeah. So being able to identify. Like if you had an extra chromosome here, then it would be trisomy at chromosome 15, right? So looking at the picture, don't get mixed up because we know the Y is way smaller than the X. So even though they look the different size, that would still consider normal, right? Okay. And I know they look kind of funky and bent out of shape, but it's got to be clear for the picture to be fair on the exam. Okay. This is the second unit. Can we end here?
Yes.
I'm going to end the video. It's by way too long. Da-da-da. Da-da-da.